Okay, this video is for week 10 as well of Biogenetics and Exercise. We're talking about Marfan Syndrome that, as you by now probably know, affects connective tissue and breaks it down. Um, so it is the prevalence of the syndrome is about 1 in 5,000 worldwide, which is not that rare if you think about it. We know about mutations in the FBN1 gene, which codes for connective tissue proteins. Um, so we're talking about fibrillin 1, and we'll see that later on. There's also mutations that cause the syndrome in fibrillin 2. So you see that these proteins connect to other proteins of the same type and other molecules that assist that to form a thread-like filament called microfibrillins. Um, we see that these proteins become part of the connective tissue that provides strength and flexibility to that connective tissue, which then also helps us to produce strength and flexibility, but also stability to the whole body throughout gross movement. We see that the genes are on chromosome 15, meaning once again it's autosomal, it's not sex chromosome related, okay? And it releases the amount of the functional proteins that one has to form those microfibrils. And then, of course, it leads to lesser fibrils uh, um, created, and then you have less connective tissue, or that connective tissue is defected. It's inherited in a dominant autosomal pattern, meaning that it is enough to have one copy of the defective gene to have the syndrome. Then we see that an interesting piece of information is that about 1/4, 25% of the mutations are de novo, meaning that they're not inherited, especially in the FBN1 gene. So since it's autosomal, it's naturally divided 50-50 between females and males. And also here we see no unusual division or occurrence of the syndrome between different ethnicities, which could happen in theory, but it's not happening in this case. Um, so every birth of a child to a family um, means that you have 50% chances of the mutation being passed on. Just remember, this is 50% chances of the mutation being passed on, meaning that one of the parents is at least a carrier. We see that we have several point mutations, most of which affect cysteine residues, meaning that wherever you have cysteine, a C nucleotide within the DNA or RNA and then mRNA, the active one, uh, wherever you have those two, you have problems with them within the polypeptide. Okay, um, and then that will be translated obviously into the microfibrils themselves. So the microfibrils are less stronger or weaker at those points where within the chain you have cysteine. Um, what else? So we see that the alteration or the defect is through a change to the 3D, the three-dimensional formation of the fibrils. Um, we see that usually there's no hotspots throughout the gene, meaning that mutations are spread throughout it and they're not concentrated. With the highest level of severity, we do find mutations focused on exons 24 to 32, meaning that there might be a connection between the fact that when the symptoms are severe, that the mutations are focused or concentrated in a few axons and not throughout the whole gene. Then we can talk about FBN2, which is highly homologous, meaning that they are genetically similar. To dumb it down, or to put it simply, meaning that mutations in that gene can cause similar disorders to that of FBN1. So there are a group of disorders that are somewhat similar to Marfan syndrome. It's not exactly the same syndrome, and they originate from a similar gene. So we see that the genes have about 67 axons each, and we only have 3 axons that never had, or we don't have any records of mutations occurring in them. Obviously, when you have 64 out of 67 axons that can be defective, that will give you a wide range of symptoms that are possible. Okay? And of course, you won't have a very strong genotype-phenotype correlation or relationship. 72% of the mutations are missense mutations, and you can see that other mutations include nonsense, then what we call frame deletions. It's what I taught you about changes to the reading frame, so it just moves. And then we have what we call axon skipping, meaning the ribosome does not read that certain axon by mistake. So it should have read the axon, but it's not. That's all we have for this syndrome. Once again, I'm emphasizing, if something in the presentation is not clear, please let me know.